Hello, everybody. I welcome you for the first ESID Educational Day online. Unfortunately, we cannot meet in person in Birmingham, but we hope that you will have occasion to discuss. There will be a feature that you can ask your questions during the talk, and we then have question and answer live sessions where we will hopefully have time and opportunity for a lively discussion, even if every one of you is in front of a screen and not face-to-face with the speakers. So I will share this session with Clara Franco-Jarava, who is the head of the ESID Junior Working Party. She's working at the University Hospital Valdebron in Spain. And I'm very pleased to introduce the two speakers of this morning session. First, Carola Vinuesa, who was born in Spain, where she obtained a medical degree at the University of Madrid. She then undertook specialist clinical training in the UK, where she obtained her PhD at the University of Birmingham in 2000. She then moved to Australia for her postdoctoral work at the John Curtin School for Medical Research, and she stayed. And she is now currently Professor of Immunology at the Australian National University and Director of the Centre for Personalised Immunology. Her talk will be about B-cell biology, and the title is Class Switch Recombination Occurs Infrequently in Germinal Centers. Our second speaker will be George Holländer, who is a pediatrician and experimental immunologist with long-standing interest in cellular and molecular biology of the thymus development and function. George is trained in Switzerland and the US. He is the head of the department and professor of pediatrics at the University of Oxford, but he's also professor at the University of Basel and the ETH Zurich. And last but not least, he is director of the very recently founded Bottner Research Center for Child Health in Basel, Switzerland. And he will speak about immunobiology of the thymus. So we will have these two talks and then the live question and answer session. And please submit your questions so that we can have a nice debate at the end. Thank you and enjoy this educational day.
Hello, I'm Carola Vinuesa. It's an honor to be here today presenting at ESID 2020. And I'm going to tell you two stories. The last one will be very brief, that relate to class switch recombination and the regulation of human IG. So I have nothing to disclose, and we'll get into the idea of why class switch recombination is important. We know that immunoglobulins can come in different flavors, and some isotypes are important for different functions. For example, when we have a heavy chain that is IgA, that immunoglobulin can be secreted into body fluids, which is important for immunity at mucosal surfaces. IgE, as you know, can help degranulate mast cells and basophils and not just protect against helminths, but it can mediate allergy. And we know that IgG can perform a range of functions, including opsonization, antibody-dependent cell-mediated cytotoxicity, etc. Now, this is a textbook view of a germinal center, which we know forms after immunization. And it has been thought that class switching is an important process in germinal centers after they undergo somatic hypermutation and after B cells introduce mutations in the immunoglobulin variable region genes. And this class switching is probably mediated after B cells interact with T cells in the light zone and receive cytokines that induce switching. Now, we know that class switch recombination can bring about processes that can be pathogenic. So, for example, in the context of lymphomas, chromosomal translocations can occur at the time of class switch recombination. And because it's been thought that this process occurs in germinal centers, this has also led to classify some germinal centers of germinal center origin. Now, there is also some very nice demonstration recently by Lanzavecchia that during class switch recombinations, there can be insertion of introns that can even confer cell reactivity. They can help protect against some infections, but some of these rearrangements can be pathogenic. So it is important for us to understand where exactly does class switch recombination occurs and how is it regulated. Just for the purpose of what we need to understand for this talk, I will keep it very simple. And we know that there are 3 enzymes that are required for class switch recombination: EID, ONG, and APE1. In order for these enzymes to target the switch regions, first there is an essential but not sufficient process that needs to take place, which is transcription of these non-coding mRNAs, production of non-coding mRNAs, which occur from promoters that are immediately upstream of the switch regions that need to be transcribed. So these are called germline transcripts, and they help attract this enzymatic complex to the right sites. Once they are there, they can exert these double-strand DNA breaks, and then the 2 different switch regions become juxtaposed, and the intervening DNA becomes excised. So through the process of non-homologous enjoining recombination, the new isotype or the new heavy chain can be found close to the VDJ region, and this would be encoding the new class switched antibody. Now, we have thought that class switch recombination occurs predominantly in germinal centers, and the evidence has come from multiple forms. Very early on, we learned that there was a shift from IgM to IgG, from the primary to the secondary response, and we assumed that this is because memory cells had arisen in germinal centers, and these were switched in germinal centers. We know there is less class switch recombination in mice lacking germinal centers, but of course these are genetically modified mice in which usually there is not just a lack of germinal centers, but also TB interactions are disrupted because the architecture of secondary lymphoid organs is disrupted. And also when EID was discovered, and we know the EID is essential for class switch recombination and somatic hypermutation, it became very obvious that the majority of EID is found in the germinal centers. So it was assumed that this is where both processes occurred, and it was nicely shown that in the absence of EID, not only there is far less somatic mutations, but also significantly less switching. But we now know that EID can be first expressed in cells outside germinal centers. Now, in the end, it was narrowed down to expression in light zone germinal center B cells in centrocytes due to the use of markers that are not necessarily longer used. So at the time, it was found that these transcripts are mainly produced by the DM4 fraction, identified as CD77 negative, CD38 positive IgG negative cells, which were thought to be light zone cells. But later on, we found out that this population is very heterogeneous. It contains anything from an activated B cell to a plasmablast. Now, the idea has been that class switch recombination occurs predominantly in germinal centers, but there was also evidence that it could occur earlier. So this is work from Ian McLennan's lab and Kai Michael Tollner, showing that in whole spleen sections, germline transcripts could be detected as early as day 4 at the time of TB interactions or when T cells had been recently primed. The group of Mark Jenkins also showed that you could detect IgG2A as early as 2 days after prime. And Rob Brink also showed that you could find comparable and synchronized germinal centers and extrafollicular plasmablasts that both contain switched cells. So we know it can happen early, but the idea has been that it predominantly occurs in germinal centers. So does this hold up? Do we think this is a site where it is favoring class switch recombination? So for this, the first thing that we did, and this work was done by Jonathan Rocco in my lab, was to look very early on after in a mouse model where we can very easily track germinal centers and extrafollicular plasmablasts. So these are switch HEL mice in which B cells carry a knocked-in B cell receptor specific for the protein HEL. They are immunized by with a conjugate of HEL and Schieber blood cells, and HEL has been mutated, so the interaction is of not such high affinity. And these are transferred into a recipient congenic mouse. And by flow cytometry, we can use a series of markers to separate from the very earliest stages the germinal center from the extrafollicular plasmablast response. And this is true for we can use any combination of markers and we always get the same result. And we can see that up to day 3, and this is important, we do not see two populations. It's only clearly from day 4.5 or between days 3.5 and day 4.5 that we can start differentiating a germinal center from an extrafollicular plasmablast response. This is also seen by immunofluorescence. So in the first 3 days, the HEL-binding B cells are at the TV border or entering the follicles, but there is no germinal centers between days 3.5 and 4.5 when we clearly start seeing these cells accumulating. So I think this is important. Remember that before day 3, there are no germinal centers, and they are clearly seen by day 4.5. So if we now ask when are these germline transcripts produced, this is now gamma-1, which is the predominant isotype in the response to HEL-Schieber blood cells. And we can see that the gamma-1 germline transcripts peak on day 3, and I told you there are no germinal centers on day 3. And by the time the germinal centers start forming, there is this sharp decline in switch transcripts, suggesting that the initiation of switching occurs prior to germinal center formation. We get a very similar picture for gamma-2b, with the peak between days 2.5 and 3, and a clear decline when the germinal center is starting. If we now look at expression of EID, we can see that there is EID produced prior to germinal center formation. So we clearly see a signal at day 2.5 and day 3, with a peak at the time when germinal centers start forming. And we get a very similar picture of BCL6, no BCL6 at day 3, when we already are seeing germline transcripts peaking, and BCL6 highly expressed thereafter. And this is, as you remember, the transcription factor for germinal center B cells. So we asked whether we could be missing a key switching event, and because we were doing bulk cells. So we looked at single cell, and we did single cell PCR at day 3, and then during the germinal center phase at 2 different stages, the dark zone or B cells and the light zone B cells. And we sequenced 100 light zone B cells. And what we found is that pretty much as we've shown before, most of the switch transcripts, practically all with some exceptions, are produced in blasts by day 3, with very few events observed in the germinal center cells. Whereas if you look at EID, we can see expression both before and during the germinal center reaction. And if we look at BCL6, it's clearly expressed in germinal centers. So again, this suggests that there is a very small fraction of B cells that appear to trigger switching in the germinal center. So the next question is, where is class switch recombination activated? So for this, we took advantage of a mouse model in which upon formation of a gamma-1 switch transcript, the B cells turn from red to green. So green cells are those that have already produced a gamma-1 germline transcript. And in these mice, we already see cells starting to switch at day 2 at the TV border, so before there is any germinal center produced. We can also see that by day 3, many of these cells have proliferated and are now extensively occupying now some follicles and moving to the bridging channels. And by day 4, we now see these green cells both in extrafollicular foci and germinal centers, suggesting that there is no predilection for switching in germinal centers. And you can see how very nicely by day 5, we get abundant extrafollicular and germinal center cells that have switched, probably deriving from these early progenitors. Sorry. Now, I've shown you data for RNA germline transcript, which is essential but not sufficient for class switch recombination. what if we look at the protein, at the antibody itself by FACS, and we see that as early as day 2, we can already detect IgG1 on hell-binding B cells. What we also compared is the fraction of cells in either extrafollicular plasmablasts in green or germinal centers in purple that have switched at any one time, and at no time we found higher switching in germinal centers than in the extrafollicular response. In fact, later on, it might be the opposite. So no evidence that switching is favored in the germinal centers, and we've replicated this with different antigens, different adjuvants in longer-lived germinal centers and always obtained the same results. So we could also ask, do we see an increasing rate of switching during the different phases of the germinal center response? These are the kinetics of a normal germinal center reaction, peaking around day 8. And we can see that from this day 8 onwards, we do not see an increase in the fraction of switch cells, again suggesting that switching is not ongoing in germinal centers. So we then ask the question, so can we identify the timing of class switch recombination relative to somatic hypermutation? So does it occur before or after mutation? And what we also wanted to do is to be able to look at a polyclonal response, or rather than looking at a B-cell transgenic response as we've been doing previously. So for this, we collaborated with Gabriel Victoria and Luca Messin, and we used his or their photoactivatable GFP mice, so we can shine a light on an individual germinal center. This makes the germinal center B cells change color, and these cells can be sorted, and we can sequence their heavy chain and their VDJ region to look for somatic mutation. And this is a representation of the clones that were chosen to be sequenced and analyzed. So each of these columns is a germinal center. Each of these boxes is a clone within a germinal center. In green, we have IgM. In gray, we have IgG. So for the analysis, we chose clones that every clone that contained both IgM and IgG, since we were interested in switching. And we enriched for clones where there were at least 4 IgM cells to increase our chances of see a switching event. And what do the results look like? Well, this is just a legend for you to understand how we are going to look at the data. In the x-axis, we are looking at the number of mutations. And then in white symbols, we have IgM, black IgG1, and colors are other isotypes. And these triangles represent the predicted class switch recombination point. If it's not filled, it means that in that particular clone, there wasn't a class switch recombination event. So for example, here we've got the predecessor of an IgG1 and an IgM cell, so the class switch recombination occurred at that spot. So what do the data look like? So this is the snapshot of the clones 15 days post-immunization. And we can see, if you look at where these triangles are located, that they predominantly occur at 0 somatic mutations, with some clones never switching, and a couple of exceptions of a 1 mutation and a 4 mutation. And this is shown here, quantified. So the vast majority of events occur at 0 mutations. Now, at day 20, different experiment, you can see exactly the same thing. Most class switch recombination events occur at day 0, and we have some unswitched. Again, showing you that, if anything, class switch recombination occurs prior to the onset of somatic mutation, and therefore it's likely to occur prior to the germinal center. Also showing you that some germinal centers are really dominated by IgM, and this indicates that the probability of constant switching is negligible. So we also looked at the enzymes that are mediating class switch recombination. APE1 is critical for class switch recombination and not so much for somatic mutation. And we found that actually APE1 is downregulated in germinal centers, which we thought it was quite interesting. And we did find a report in mice showing APE1 protein also low in mouse germinal centers. We confirmed this in humans, so both dark zone and light zone cells have decreased the amount of APE1 expression. And we confirmed this by immunofluorescence, and you can see how APE1 is largely absent from germinal centers, whereas APE2 is predominantly expressed in germinal centers, and APE2, as you know, is required for somatic mutation. So there seems to be a distribution of labor in that switching does not occur in germinal centers, or largely does not occur, whereas somatic hypermutation is favored in germinal centers. Now, intriguingly, we speculated that perhaps BCL6 was repressing APE1, and we looked at this chip-on-chip data produced by Jose Polo, and indeed we could see that BCL6 binds to the APEX1, it's enriched at the APEX1 promoter, but not at the APEX2 promoter, and these are controls that had already been known to be enriched for BCL6. So this is the summary. Class switch recombination occurs early during the first TB interaction at the TB border and prior to germinal center formation. And we're very happy. This was published in Immunity, and with this beautiful cover by an Aboriginal Australian artist. And we're also very honored that Immunity chose it as one of the best papers of 2019. And this is Jonathan Rocco, who did all this work. Sorry. So I just want to finish off by telling you a brief story that I think is also quite interesting related to class switch recombination. And this has to do with regulation of IgE, which, as you know, can mediate allergies and atopy. So we've looked at human tonsils, and human tonsils are quite interesting. They are exposed to a lot of innocuous antigens like food in itself, but also to allergens, food allergens, inhaled allergens. And if you look at the population that normally contains TFH cells, so PD-1 high and CXCR5, we find a population that we would have normally classified as Tregs because they are CD25 high and CD127 low. But these Tregs, unlike the Tregs in the other fractions, do not express FOXP3. So you can see that both as a percentage as MFI, these CD25 positive follicular T cells do not express FOXP3 in the tonsil, or largely. There are some, but they are a minority. Another interesting thing is that largely these CD25 positive follicular T cells are methylated, which suggests that they do not have a thymic origin. They might have been induced peripherally. And if we look at the RNA transcription profiling, and this is also seen at the protein level, they largely resemble Tregs in practically everything except expression of FOXP3, which is not very high and comparable to naive T cells. So they have the highest amounts of CTLA4 in the tonsil, even more than Tregs. They express high amounts of GTAR, IL-10, low amounts of CD40 ligand. And we tested their suppression capacity, and you can see from this graph that these FOXP3 negative, largely CD25 positive TF cells, are as suppressive as conventional Tregs, whereas you can see that conventional TFH cells do not suppress the proliferation of naive T cells. So they look like Tregs, they behave like Tregs, they do not have FOXP3, and one of the things that really distinguishes them from other T cells in the tonsil is that they express most of the IL-10 expressed by T cells in a tonsil, even more than Tregs. So this is quite striking. And what Pablo showed is that if you look at IgE induction by TFH cells, if you now add these CD25 positive T cells in the cultures, they largely repress IgE, whereas they do not repress IgG. So what are these CD25 positive TF cells doing to IgE? So what Pablo showed is that they actually are repressing this epsilon germline transcript, which is the first step in switching to IgE, but they do not change gamma transcripts. And Pablo showed that this in fact is mediated by IL-10, because if he now blocks IL-10 with an anti-IL-10 receptor antibody, this repression is lost. So the final slide is showing you that there seems to be a very interesting negative correlation between the amount of these follicular CD25 positive T cells and serum IgE in children. So in a collection of matched tonsil and blood samples, he showed that those children with lowest percentages of CD25 positive TF cells in the tonsil had the highest amounts of serum IgE. And this is true whether we look at this fraction out of Tregs, out of total CD4s, or whether we only focus on the IL-10 positive fraction of these CD25 positive TF cells. So the lowest amounts of these IL-10 positive cells, the highest the serum IgE. And this is not true for total FOXP3 positive T cells, or for total IL-10 positive CD4s, or for Tregs. So this specific subset seems to be quite important in repressing serum IgE. And this is very similar where we've separated children that have high IgE from children that have low IgE. And those that have high IgE tend to have a lower fraction of the CD25 positive TF cells. So in summary, we'd find that in mucosal sites, in tonsil, that is exposed to allergens, we get these CD25 positive TF cells, which might be peripherally induced, look like Tregs, repress like Tregs, and specifically repress switching to IgE via production of IL-10, and this leads to lowering of serum IgE. So again, this was published in JXMed, and we are also very proud and happy that it was selected as one of the top 10 papers last year by JXMed. So with all of this, I've thanked the people that contributed to the work, but I would also like to thank most of the members of my lab that have helped in this work. I've mentioned the collaborators as I was going ahead, so this is all. Thank you very much for listening. I hope if you have any questions that I can answer them. Thank you.
Good morning. My name is George Holländer. It's a pleasure to present to you some of the basics of the immunobiology of the thymus. For this lecture, I have a faculty disclosure shown here, and would like to provide you now with an overview of the immunobiology of the thymus. As you know, the thymus is a bilobulated organ in the anterior compartment of the chest and sits on top of the heart. If one were to cut through one of these thymic lobes, the histology would reveal that we have an outer cortex, cell dense, as shown in this slide, and an inner medulla, which is less dense. Now, what is the role of the thymus? The thymus contributes in essential ways to the capacity of the immune system to distinguish between self and non-self. Let us do a short experiment here. You know this lady rather well, and if you look a little bit closer at her, then you will realize that something has changed. Your cognitive recognition of a different pattern is exactly what the immune system does at the molecular level, being able to distinguish between self and non-self. Self is to be defined mechanistically and functionally as to be tolerant to, while the non-self array of antigens should provoke a directed and effective immune system. In the center of this capacity are T cells, which originate from the thymus. So let us look a little bit closer at what means self and non-self. The concept of self is defined, obviously, by the genes in our genome, and for the human, we're talking about 21,000 protein coding genes with the possibility to produce an endless array of different proteins. This equates to the blueprint for over 4,000 different cell types. In our adult body, we have 3 times 10 to the 13 of these cells. And within each single cell, we have again up to 1 billion proteins and an enormous number of cell divisions over a life course. All this will need to be recognized as self and tolerated as self, i.e., no response of the immune system against these molecules, cells, and tissues. In contrast, non-self, obviously, is the whole array of microbial agents, including bacteria, viruses, fungi, protozoa, helminths, but obviously also altered self, such as malignantly transformed cells. Here we are talking again in enormous numbers that the immune system should be able to recognize and respond properly to. We have a couple of tens of millions of different animal species. We have in the same magnitude different virus types. And for bacteria, there is an estimate that there are approximately up to 1 billion different bacteria. So, if the task is for T cells to make that essential distinction between self and non-self, where do they learn this capacity? It is, as already alluded to, the thymus, where this principal competence is instructed. And the thymus, as you will see, is an organ that, though shows the first signs of senescence-related reduction in size and output, the thymus nonetheless can still be found as a functionally competent organ in octogenarians and older individuals. So what happens if, as shown here on the left, a uncommitted T-cell precursor enters the thymus so that at the end of a journey through the thymus, mature T-cells can exit? This is a process that is age-dependent, and as already alluded to, the age defines the thymus size. So in individuals at around 1 to 2 years of age, the thymus has the highest number of cells per total body weight. Then later in life, and after puberty, the organ is clearly decreasing over time. This decrease affects exclusively the parenchyma that produces T cells, with a decrease both in cortex and medulla over time, and adipose tissues infiltrating the perivascular space and eventually taking over and leaving only small islands of thymopoietic activity behind. In that capacity to produce normal T cells, the precursors enter the thymus at the junction between the outer cortex and the inner medulla. These precursors then travel through the depth of the cortex to an area under the cortical capsule, only to return from there, bridging the corticomedullary junction, and to access the medulla, and eventually, after a journey of a couple of weeks' time, in the mouse approximately 3.5 weeks, in humans this takes much longer. Until that journey, they exit through the blood vessels to the periphery. During this journey, several cell surface molecules allow us on the surface of developing T cells, or thymocytes, to define at what stage of their development these cells are. We have markers for the cortex and we have markers for the medulla, but most importantly here is that in the cortex, the cells start to express the T cell antigen receptor plus CD4 and CD8 to then make up the bulk of cells to be both CD4 and CD8 positive, or what we refer this to as double positive. In the medulla, these double positive cells have then matured to either being CD4 or CD8 effector T cells, or to acquire a phenotype and functional fate of NK T cells or T regulatory cells, if we are talking about, as shown here in this square, the T cells with an alpha-beta T cell antigen receptor, and then of course also in the medulla we will find mature gamma-delta T cells. Importantly, this progression from a uncommitted hematopoietic precursor, here shown on the very left, to a mature functionally competent T cell, shown on the right, to exit, this competence is not cell autonomous, but is instructed by a very complex microenvironment that builds physical contacts and functional interactions with the developing thymocytes. And what you see here in this codex image is the intricate three-dimensional lattice formed by thymic epithelial cells. These thymus epithelial cells can be differentiated, as you will see, into outer cortical and inner medullary epithelial cells. And some of the medullary epithelial cells even express the transcriptional facilitator AIR. In addition to these epithelial stromal cells, you will find dendritic cells, B cells, macrophages, endothelial cells, and fibroblasts that all have a very specific function and contribute to the generation of this unique microenvironment able to support T-cell development. And if we now project onto this T cells, for example, that express CD8, then you can see that these CD8 cells in the in the cortex are very crowded, and typically they also express CD4, and that the medulla is less cell dense for thymocytes. The two different thymic epithelial cells, namely the cortical cells and the medullary cells, have completely different functions, important for the different steps in intrathymic T-cell development. Cortical thymic epithelial cells attract hematopoietic precursors from the blood, commit them to a T-cell fate, and allow their expansions to immature thymocytes. It is then that two selection processes take place. The first one, called positive selection, which secures that T cells express a T cell antigen receptor that can recognize antigens in the context of MHC. And the second selection step, referred to as negative selection, makes sure that none of the T cells that have a functional T cell antigen receptor recognize with high affinity self-antigens. The medullary thymic epithelial cells provide a second line of negative selection, induction, and then allow a post-selectional maturation of those thymocytes that have survived, and finally also create the niches that allow regulatory T cells to differentiate and exit the thymus. So, how can we further distinguish the different thymic epithelial cells in view of the fact that they realize such a broad, different array of functions? One way is to use flow cytometry, and with that tool, we can identify easily cortical, mature and immature, medullary, mature and immature, as well as senescent or geriatric medullary epithelial cells. At the moment, the concept based on flow cytometry would argue that there is a common precursor of epithelial cells that give rise to cortical and medullary epithelial cells. A second way to look at the different thymic epithelial cells and their developmental past is to use single-cell RNA sequencing. And when we analyzed thymic epithelial cells with this particular platform, we realized there are at least 9 different thymic epithelial cells that are defined, on one hand, by their particular gene expression profile, as shown here, where you have on the right, in color code, the different clusters, and along the bottom of this heat map, particular genes that identify these different cluster types. With this approach, we can identify cells such as the perinatal cortical epithelial cells, which appear to be the precursors, at least early in life, to all thymic epithelial cells. We can identify mature cortical epithelial cells, as well as immature and mature medullary epithelial cells. The analysis also revealed that there is a big population of epithelial cells that falls somewhere, as defined by the gene expression profile, between cortical and medullary epithelial cells, and we call them the intertypical TEC. Now, apart from just using a new nomenclature that has a higher granularity to identify the different thymic epithelia, it also allows us over time to see how dynamically they are represented within the epithelial scaffold. And what you see here is a measurement of the different thymic epithelial subtypes over time. And you will recognize that the brown population, the perinatal TEC, which are on top of this graph, are present and easily detectable at a high frequency of up to 25% at 1 week of age, but by 4 weeks of age, they might have almost gone. And it is mainly the thymic epithelial cells that are situated in the cortex or that belong to the intertypical population that are expanding. In a similar way, we then can ask the question, how different are the pathways of these individual thymic epithelial cell populations when differentiating from a perinatal CTEC, for example, to a mature MTEC? Now, this is ongoing work that we look into using various computational biological tools to do so. But what is clear is that whatever subtype we find in the thymus, these cells are derived from a precursor that expressed the transcription factor FOXN1. FOXN1 is the master regulator of thymic epithelial cell development and consists of a centrally placed DNA binding domain, the Fourquet domain, and two laterally positioned TEC differentiation or transcriptional activation domains. When we looked at the crystal structure of FOXN1, it became clear that the DNA binding domain fits very nicely in the larger groove of the double helix, with a very small, interestingly limited DNA motif to be recognized at this interface. Now, this is particularly interesting because if one were to change that DNA binding, the function of FOXN1 is altered. So let us first look at the normal regular function of FOXN1. FOXN1 plays a role in the homing of blood-borne lymphoid precursor cells. It is important for their commitment to the T-cell fate. It is essential for their expansion, but also the positive selection of immature thymocytes. It plays indirect roles in negative selection, be it in the cortex or in the medulla, and FOXN1 again is essential for the program that allows post-selectional maturation of these cells. So FOXN1 is clearly very central to the whole process. And as I already alluded to, FOXN1 is expressed from the very early precursors in the embryo to the phenotypically mature thymic epithelial cell subsets. If FOXN1 is missing because the Fourquet domain can't bind to the DNA, then there is no T-cell epithelial, there's no thymic epithelial cell development, and the result is the so-called nude mouse, which is nothing else but a deficiency in FOXN1 function. And as you may know, they are patients with the same genetic pathology that lack a thymus. Now let's go back to having a full complement of normal thymic epithelial cells. And these interact with developing T cells. And the developing T cells are marked by the fact that they first rearrange their beta chain and eventually their alpha chain, and by doing so, create a complete T-cell antigen receptor that is first fully expressed at the double positive, so CD4 and CD8 double positive stage. If we look at that T-cell receptor through a plane and now look from above, then you can see that what the T cell receptor recognizes is the groove and the two rims of MHC molecules that form the fold for the antigen to be positioned and presented. And a better structural understanding of these antigens and how they fit into particular MHC molecules has helped us to understand the structural constraints of this interaction. Now, what is the quality control of the TCR specificity? Because as said, these TCRs are chosen completely randomly, and we need to make sure that they are of a particular usefulness to an individual. And then again, what are the antigens that are being seen in the thymus? So if we look a little bit closer at the selection process that I already alluded to, we have learned that positive selection is usually a brief and infrequent signaling limited event between thymic epithelial cells in the cortex and the developing thymocytes. And this interaction takes only a couple of minutes. The result is that a transient TCR signal is created, which allows the cell to survive. So it's basically the survival signal. During negative selection, which takes place in the cortex and the medulla, the interaction is, because of the higher affinity of the TCR, extended, longer. And if optimal, the cell stops migrating, and this extended TCR signal results in cell death, because the system has proven that the T cell receptor is very reactive to MHC plus self presented in the thymus. If that signaling is only suboptimal, the cell has basically a second chance to get out of the path to die and potentially rearranged, for example, the alpha chain to make sure that it expresses now a T cell antigen receptor that can escape negative selection. If we look at the processes of selection from a point of T cell receptor affinity, then you can see that the low affinity does not provide the survival signals, and thymocytes will die, here on this red curve on the left. If the affinity is within the range of normalcy placed, then there is positive selection and survival. And if the affinity is much higher, then the cells are, though positively selected, but will be negatively selected later on as they provide a certain danger for cell death. The thymocyte lineage decision: why is a double positive cell basically turning into a CD8 cell that is recognizing MHC class 1, or into a CD4 cell that recognizes exclusively MHC class 2, is again provided by a set of signals that, in the event that the TCR signal ceases, allows the adoption of a CD8 single positive phenotype, whereas if the signal persists, CD4 single positive cells are generated. Lastly, just to show you that the process is rather complex, we have in the cortex not only the generation of alpha beta T cells, but also NK T cells, which then in the medulla are subjected to different fates, either as an NKT1, 17, or 2 NKT cell, and this is again dependent on the TCR signaling strength. Now, last but not least, what are these developing T cells seeing in the thymus while they are developing? They see a representation of the molecular self. And this is not only a representation of all the organs in our body, but also an anticipatory mechanism that allows to foretell what kind of antigens might be seen at different cell stages, at different ages of an individual. So T cells in the baby already anticipate post-puberty tissue changes. This is because thymic epithelial cells can express up to 92% of all protein-coding genes. And they do that particularly efficiently in the presence of the transcriptional facilitator AIR, which, as shown here, is responsible for an expression of a subset of approximately 4,000 protein-coding genes. And this promiscuous gene expression affects particularly loci that are usually silenced due to epigenetic marks. So AIR is absolutely essential that we can generate a repertoire of self that represents a library on which T cells are educated. Now, the question finally is, are these cells in the medulla expressing all protein-coding genes, or only a certain pattern, or is that expression random? And to answer this question, we use single-cell transcriptomic analyses of medullary thymic epithelial cells. And what you can see is here as a final result. It is a random process that represents in individual thymic epithelial cells our self, and AIR-dependent tissue-restricted antigens, or TRAs, are a couple of tens per single cell. AIR-enhanced TRAs are a couple of hundred, and the other tissue-restricted antigens that are expressed by thymic epithelial cells are expressed in a fashion independent of AIR. So overall, we have few thymic epithelial cells that express a particular tissue-specific antigen, but this antigen is usually expressed at a relatively high frequency. So T-cell tolerance then is mediated in the wild type by negative selection, that if it's a very high affinity creates clonal deletion. If it is a not so high but still high affinity, creates an alternative fate with regulatory T cells being generated, thus complementing recessive central tolerance with a dominant central tolerance. And in the absence of AIR, what we end up is obviously with an escape of forbidden clonotypes because certain antigens have not been used or were not available for negative selection. And so in the end, APS1, the deficiency of AIR expression, obviously results in a number of autoimmune pathologies because T cells were not properly educated. Primary lymphoid organ is therefore, the thymus as a primary lymphoid organ is therefore the site for generating naive T cells with different functions. It has a complex stromal scaffold that controls lymphoid development and selection, and is also the site where a molecular representation of self assures that we can establish efficient central tolerance. Thank you very much for your attention.
So I would like to thank the two speakers for their fascinating talks. We have several questions that have been addressed from the audience, and I would like to start with the first speaker, with Carola. Your results that class switch recombination actually occur prior to germinal center formation and prior to the onset of somatic hypermutation alters our current understanding of antibody responses. And I think that insights into the regulation of this mechanism is very important, as the dysregulation can cause cancer and autoimmunity. And there's a question from Marta Rizzi for you. She asked, extra follicular class switch occurs when AID is still very low in expression. Is there another mechanism promoting class switch?
No, we think that those levels might be sufficient. So it clearly is expressed and it's positive. And it might be that low levels are enough for class switch recombination, whereas higher levels are required for somatic hypermutation. And it might be that it's the combination of enzymes that it works with that it's critical. So APE1 would act for class switch recombination and APE2 for somatic hypermutation. The expression is very clear both at the RNA and protein level. So again, we think it might be sufficient.
Thank you. Clara, you want to join for the questions?
Yeah, sure. There were a lot of interest for the second part of Carola's talk, so I combined some of the questions. So the first one would be, is there any work on the specificity of CD25 positive T regulatory cells, TCRs, in the tonsils? Are they found elsewhere, like in Peyer's patches? And finally, what is the effect of these cells in children? Do children with high IgE and low CD25 positive T follicular helper cells have food or other allergies? I can repeat the question.
Thank you, Clara. There hasn't been any work so far on the specificity, and as you know, working with human tonsils is different. It's difficult because each child has a different background, and we can't follow or track antigen-specific T cells. So it's an important question, but there is nothing done really to date. In terms of the food allergies, children that undergo tonsillectomy tend to have a higher frequency of allergy and atopy, so that has been described. Now, our sample was relatively small. We matched 50 blood samples to tonsil samples. So it is true that we had some allergic children in the mix, but I think the sample is underpowered to establish a real correlation between allergies and this high IgE. We definitely had a very nice correlation, as I showed, between having low follicular CD25 positive T cells and having high amount of IgE. But the correlation with allergy is would need a more powered sample. I think you did ask something else, but I don't know whether I...
Yeah, sure. Yeah. I asked if these cells are found elsewhere, like in Peyer's patches.
Yes, look, they are very prevalent in all mucosal surfaces, so they are in Peyer's patches. We also found them in ordinary lymph nodes removed during, for example, colon surgery, so pelvic lymph nodes. And in those lymph nodes, there are a smaller fraction of the Tregs in the lymph node. So we think they are expanded in surfaces or in lymphoid tissues that are exposed to environmental and inhaled and food allergens. So that's why we think it might be some type of induced peripheral Treg that enters the follicle. But we do find them, as I say, in most human lymphoid tissues, but they are more common in tonsil and in other mucosal-associated lymphoid tissues.
Christina, do you have questions? You have to unmute.
Maybe we switch to George. I had to activate my mic. And so there are two persons in the audience, Kate Sullivan and Efronisi. I can't speak out her name correctly, sorry, who ask how long does it take a nascent T cell to develop in the human thymus? Because you said 3.5 weeks in mice, and people here are interested in human.
Yes, of course, and I do understand the question. I hoped that I could get away with it without providing a very precise answer, because it is more difficult than obviously under experimental conditions. There is no track and trace, so to say, for human T cell development except under the conditions of bone marrow transplantation. And there the observation is that the speed of recovery and hence the detection of mature T cells in the periphery is also correlating to the age of the transplanted patient, in addition to a number of other confounding influences, such as the disease nature and the conditioning of the patient. So I think for humans, we can assume that the younger, the faster, the more complications such as graft-versus-host disease, radioablative or chemotherapy, that they all delay the recovery. And so T cells can be found in uncomplicated young individuals transplanted within a couple of months. In adults, it can take up to 2 years to reach normal, for example, CD4 levels. So that is very variable. What we also know from animal experimental conditions is that these 3.5 weeks can be actually shortened under certain conditions where there is an absence of GVHD, obviously, but where growth factors are provided. So the speed by which a precursor differentiates into a mature T cell is not cell inherent to the hematopoietic system, but is really the result of a crosstalk between these two.
And just to follow up with this, from your experiences, would there be some external factors that could help speed up the immune reconstitution after transplantation?
In the human system, as you know, various thoughts have gone into this, and from the experimental work, things like keratinocyte growth factor or IGF were being considered, but none of those found use in the clinics, possibly because of their in-part side effects, such as KGF was always thought that given too long would actually result, after an initial boost of thymopoiesis, in a contraction of the thymic microenvironment and hence lesser output. But to my best of knowledge, there's nothing in the clinic in use that has translated sensibly from experimental work that looks a little bit more promising.
Thank you. Clara?
Yeah, we have another question for George Holländer, which is that why is that in air deficiency mostly endocrine autoimmunity appears and not much a wider range of autoimmune diseases?
Very good question. A question that still puzzles us who are interested in promiscuous gene expression and in the role of the way how self is represented in the thymus. Indeed, endocrinopathies are the main feature of air deficiency, and the clinical presentation is very striking as it affects mainly endocrine organs. On the other hand, we also know that non-typically endocrine organs, such as the lung, the prostate, can also be targeted as part of the pathology that ensues consequent to a lack of air. Why we see these organs and, for example, not other autoimmune pathologies of the musculoskeletal system or even of the central nervous system remains unclear. There is probably some indirect explanations, I think, that one could consider, and that concerns the fact that the air system is backed up by other mechanisms that will help to induce and maintain peripheral T cell tolerance. And those are air-independent mechanisms in the periphery, and probably they are equally well for some, but not for other organ systems to protect and cause T cell tolerance. But why particular endocrine systems are susceptible to this, I think, is clearly unknown.
Thank you. And related to this, could we have like APS1-like phenotypes if we had effects on other facilitators than air?
Yes, so there is the second molecule that has been considered, which is called FESF2, as a regulator of promiscuous gene expression with non-overlapping targets, or largely non-overlapping targets that are promiscuously expressed. And at the moment, the thinking is that probably FESF2 and AIR collaborate to a certain extent, but This is murine findings, and to my best of knowledge, there has not been a single patient with a FESF2 deficiency described yet. Now, 4,000 cells have been identified in the mouse to be regulated by the presence of air. We have many more tissue-restricted antigens that need to be sensibly represented in the thymus and are represented in the thymus. And so we expect that there are other mechanisms that play a role than just air for the phenomenon of promiscuous gene expression. And wherever you have a molecular mechanism that is important, the suspicion should also be that there is a disease that occurs in the absence of that particular mechanism.
Thanks a lot. So I see there are lots of questions. Maybe, Despina, you could pick up some in the meanwhile.
I will ask a question to Carola from Arnon, who asks, since class switch recombination and somatic hypermutation occur before B cells enter the germinal center, is there another known role for AID in the germinal centers?
Look, I think there's a few reports showing some level of somatic hypermutation before the germinal center, but we have to remember that this is a very low level compared with what occurs within the germinal center. So I think the classical view that AID is important for somatic hypermutation in the germinal center is correct. The germinal center is an enormous source of mutation. There is continuous, ongoing mutation that increases the affinity of the B cell receptors throughout the germinal center response. And I think that there is very good evidence. So my view would be that no, the main role of AID is in the germinal center, is to facilitate somatic hypermutation. And there is relatively limited amount, with some exceptions. There could be some infections where this occurs at a greater magnitude, a little bit of evidence perhaps in malaria, salmonella, but clearly for most T-dependent antibody responses, somatic hypermutation occurs predominantly in the germinal center thanks to the actions of AID.
And I would follow up with a question for myself. What is your view? May the timing of class switch recombination that is occurring prior to the germinal center reaction have an advantage with regard to the host response to infection?
Look, I think, of course, the host response to infection is not only the germinal center response. There is a very rapid extrafollicular response that occurs and is very protective. And this is important, particularly also in the context of T-independent antigens, or antigens or responses to bacterial pathogens that divide very rapidly, where an early response could mean the differences between, life and death. And some isotopes are more efficient, we know, at opsonizing and, causing complement activation. And switched isotopes tend to be better. So I think there is important roles for class switch recombination prior to the germinal center response. But I think it's also I think the importance of class switch recombination occurring prior to the germinal center reaction and not within the germinal center is to always preserve an unswitched compartment so that we can increase diversity in the face of a second infection or an infection with a related but not identical antigen, against which different isotopes might be more useful than the isotopes produced during the initial response. So the beauty of the immune system is that by all its means in continuously diversifying, it wants to keep options open. So to prevent switching in germinal centers is the only way to maintain an unswitched population in the memory compartment that can decide to switch to other isotopes in the face of a mutated antigen on a second round of infection.
Thank you.
So we have a question from Kate Sullivan to Carola. She says that it's a lovely work, and then she asks, if you think that class switch recombination occurs differently at mucosal sites leading to IgA.
Look, in our work, we did do some mucosal sites and IgA switching, and we observed exactly the same thing. We have it in the supplementary data of our paper. It occurs independently and outside germinal centers. So we think this is probably a general mechanism whereby germinal centers turn off the ability to undergo class switch recombination. I'm not saying that the cells that switch are identical in mucosal and in, gut tissues than in other secondary lymphoid organs, but I think it's probably a generalized phenomenon that in germinal centers, wherever they are, whether it's IgA to IgE, switching is relatively inhibited in germinal centers.
Thank you. And then at the context of a host response to infection, which advantage or biological role will have this fact of having class switch recombination before the germinal center creation?
Look, this is a little bit like the question that I just responded to Despina. I mean, on the one hand, not only the germinal center response has to be switched, the extrafollicular response, if it's switched, it can be much more protective. And we know, for example, in the context of bacterial polysaccharides, to, let's say, Haemophilus influenzae, Streptococcus pneumoniae, Neisseria meningitidis, these are T-dependent antigens. They will not undergo a germinal center response. And switched isotypes are more protective because they fix complement, because they can engage a number of other, effector cells that will help opsonize the pathogen. So having an extrafollicular response switched is important. But also, as I said, I think it's equally important not exhausting all our switching options. If switching occurs in germinal center along a class switch along a germinal center response, we would soon exhaust. There would be no IgM cells left. That means that during a memory response, we wouldn't have the choice to reverse back and have, IgG subtypes that we haven't, generated in the primary response. And who knows if we encounter a related antigen or a virus that has mutated since the first encounter, which is typical. We know that even viruses that don't mutate a lot mutate from season to season. We are hearing that with COVID, with flu, so we want to, and each version of the virus might need to recruit a different type of effector cell. So I think there is enormous evolutionary advantage at preserving diversity. And the only way to preserve diversity is to stop switching at a time where B cells undergo massive proliferative expansions and continuous rounds of turning on, somatic mutation, etc. So I think it makes sense from both points of view.
You will switch again to the thymus. George, there were some questions. There was Tim News who wants to know if innate lymphoid cells visit the thymus.
Sorry, I didn't get the question at the end. If innate lymphoid cells
visit the thymus.
Visit the thymus, yes, they do. We can recognize ILC2 cells within the thymic microenvironment at various stages of development. Whether and to what extent they have an inductive nature remains untested at the moment. But I would think and speculate if they are there, they have a role.
And in line with this, there's Catherine who wants to know if NK cells, if you can elaborate on NK cells and their education in the thymus and subsequent diseases.
So I'm not really the expert here on NK cells, but NK cells are generated in the absence of a thymus. Hence, their existence does not require a thymus, even if they might transit at one point through the thymus as described. So the education, if there is any for NK cells, is clearly thymus independent.
Okay, thank you, Clara.
We have a question from Andrew Kennedy, also for George Holländer, and says, can we differentiate between a recent clinic immigrant that emerged a week ago versus one that emerged years ago but has not met antigen, like a new or old recent chemical?
So the question is, can we, if I understood it correctly, can we distinguish the age of T cells as they have exited the thymus with regards to the time when they were generated?
Yes. Okay.
So there are various tricks that people have tried, be it cell surface markers, be it excision circles generated after recombination of the TCR locus. The only phenotypic, molecular marker that appears to be significantly informative are the TREX, although as soon as you have post-lymphoid peripheral depletion, a massive expansion of T cells, also that marker will be diluted independent of the age, since these cells have been generated. So the only way at the moment that we know about the ages and the behavior of cells dependent on when they have left the thymus is under experimental conditions, for example, where you can mark the cells that exit the thymus with a fluorochrome, and the expression of that fluorochrome decreases over time. And you can then stage the age of the cells according to the remaining expression of that fluorochrome. And good insight has been gained from these experiments, but how they translate to the human and to complex situations of, let's say, T cell repertoire reconstitution is not known to me.
Thank you.
So I would like to continue with a question from Fatima to Carola. She asks, have you studied effect of tonsillectomy on class switch recombination to IgE and development of allergies?
Thank you, Fatima. No, look, we haven't studied it. My guess would be that other secondary lymphoid tissues might take the role of the tonsils. Now, we have adenoids and other mucosal-associated lymphoid tissues, but it'd be worth testing. It's difficult because we use the tonsil as the easiest secondary lymphoid organ to obtain from a human. So I'm not sure that it'd be so easy to obtain additional tissues, but I would guess, since we find these TFR cells practically in every human secondary lymphoid tissues, although they are very abundant in tonsil, but they're also abundant, as I said, in other mucosal-associated lymphoid tissues.
Thank you. Clara?
Yeah, I'll get back to George Holländer, and a question from both of you. What is the role of NF-kappa-B2 signaling in the thymus, as these patients also have endocrinopathies?
Bodo, very good question. Thank you for asking this. Knockout experiments in the mice have started to look at various pathways and how they affect thymic epithelial cell differentiation. And if you delete NIC, which is a kinase upstream of activation of NF-kappa-B, then what you see is that medullary epithelial cell differentiation is impaired. And consequent to that impaired medullary epithelial differentiation, a normal medullary scaffold of epithelial cells is missing, and education, and in particular negative selection, is impaired. So T-cell receptor repertoire deficiencies, in the sense of the presence of forbidden T-cell clones, is a consequence of a lack of a normal thymic epithelial cell compartment. And that, regarding your question, is in part regulated by NF-kappa-B.
Thank you. Christina?
There was another short question with regard to endocrinopathies. Is there an explanation why mostly endocrine autoimmunity appears in air deficiency?
I can make it very, very short if the question is for me, and I don't know, and I think people don't know it.
Okay.
Okay, I would have one from Alfonso Rocha, and says, how many thymic epithelial cells a T cell meets or needs to meet for efficient negative selection?
So that's a very interesting question that has led to some calculations that, when further scrutinized, it became obviously untenable. We know at the moment that most likely for T cell selection in general, T cells will interact continuously over a period of time with the stromal compartment, in particular thymic epithelial cells. Whether for negative selection, you would need to have multiple of these interactions over a period of time, or whether a particular interaction suffices to induce negative selection has not been mathematically modeled. But I would think that a strong interaction of sufficient amount of length between a single T cell and a single medullary epithelial cell should suffice, for the one reason that if we look at mice at single-cell transcriptomic analyses and take this as a snapshot in time, then a particular self-antigen is expressed by a very, very low frequency of cells, 0.1 or 0.01% at a given time, but at sufficiently high concentrations. And in order to model a system where you would have repetitive interactions with exactly that self-antigen over time, the likelihood for this to occur is very, very small. So I would think in the medulla, with regards to air-dependent self-antigen expression, a single hit should suffice.
Okay, thanks. And we have another question. It says, hello, does RAC deficiency affect tissue-restricted antigen expressions by MTEX?
Yes, it does, indirectly. So there is a crosstalk between the hematopoietic system and thymic epithelial cells. And this crosstalk occurs under normal conditions continuously, and actually helps through rank-rank ligand interaction and TNF family member interactions to mature thymic epithelial cells to gain the competence for promiscuous gene expression. In the event where these mature T cells are not or insufficiently produced because of a RAC deficiency, that crosstalk is missing, and consequently the medulla cannot normally form and perform as a negative selector. And the result is a TCR repertoire that is oligoclonal, but contains a number of autoreactive T cells.
Okay, thank you. Christina?
There's another question with regard to thymic medulla from Mohammad Ibrahim, who asks, can peripheral dendritic cells enter the thymic medulla? If so, can they induce central tolerance to peripheral antigens they bring in?
So very good question, Mohammed. We can find three different kinds of dendritic cells: the classical dendritic cells 1 and 2, and then the plasmacytoid dendritic cells in the thymic medulla. The classical dendritic cell type 1 is generated within the thymus, while the two other ones appear to immigrate from the periphery to the medulla. And there, the thinking clearly is that the capture of antigens and presentations in the periphery would contribute to the mechanism of antigen presentation. The same thing is true for B cells, by the way, who also can home from germinal centers back to the thymus. And the dendritic cells that are in place are also able to profit from self-antigens that are being produced and released by thymic epithelial cells and taken up to then enforce negative selection effected by hematopoietic cells within the medulla. So the system tries to come up with a lot of different cellular interactions to make sure that the danger of escape of autoreactive cells from the medulla to the periphery is minimalized.
Thank you. So I have just a question, a little bit provocative to George. We are more and more dissecting the very molecular mechanisms taking place in the thymus. When will be the time that we can recreate individual thymic epithelial cells in the laboratory, maybe even entire organs, so that we can give them to patients with thymic defects or patients post-transplantation who have not optimal immune reconstitution?
A holy grail question, isn't it? And the question that Despina and I have discussed in the past, and I would bet we continue to discuss for a couple of years to come in the future. So there are several groups worldwide that are looking into the possibility to grow a thymus from iPS cells or from the yet to be identified thymic epithelial stem cell and form a artificial thymus, either ex vivo or eventually in vivo through a seedling, quote unquote, of the relevant cells. There are a number of very challenging issues here that one needs to recall. The first one is thymic epithelial cells, when grown in 2 dimensions in a petri dish, fail to display their magic of attracting and educating and maturing T cells. We have at the moment only a scant understanding of all the molecules and the signals that are required for a precursor to become a mature thymic epithelial cell. And with those 9 subtypes that I have shown you, we only start to understand how they relate to each other and what their dynamic development is over time. So whatever the solution will be, whether it is an iPS-derived recapitulation of development or the identification of thymic epithelial cell precursors, what also will be required for a normal thymus to be formed and transplanted is a scaffold that keeps these cells in a 3-dimensional organization and a scaffold that keeps them developing and proliferating. And the hurdles are high, but as typical, people are not discouraged by this. And we and others are working in this direction. And I hope that there is going to be such an opportunity, but it will take time, clearly take time.
So we will invite you again for another educational meeting.
Yes, do that in 20 years' time, and then we can look back on something successful, I presume.
Okay, Clara.
Thank you. So we have a final question for Carola Vinuesa, and it's from Johannes Truk, saying, how can we use your findings to better understand functional deficiencies in patients with antibody disorders?
Look, I think, what I've shown you is something very fundamental, and I think sometimes, we've equated switched isotypes or the lack of switched isotypes with problems in germinal center reactions. And what we've shown is that it really has very little to do with a germinal center, that switched isotypes can occur independently of germinal center reactions. So, it doesn't change much in that a lot of our, hyper-IgM syndromes have still got to do with problems with AID and CD40 signals. But I think it's a conceptual shift, not just for antibody deficiencies, but also for cancers. We've thought that, a cancer that mainly is composed of switched plasma cells or switched cells needs to derive from a germinal center. And I think this really changes our understanding to think that switching is independently of germinal centers. And so I don't think it will really change the way we look at patients with antibody deficiency, but I think it changes our understanding of why and where switching takes place, and why is it important mainly to have unswitched memory B cells for protection. So I'm not sure if I'm answering your question, because I don't really think I'm going to change the management of antibody deficient patients, but I think it's interesting to think conceptually at the significance and the implication of not having switching occurring in germinal centers.
Okay, thank you very much. So Despina, if you want to close.
So thank you indeed to the speakers, to all the participants all over the globe who asked questions, and we were able really to address all the questions we got in this discussion. And I hope that you enjoyed this meeting, even if it's quite weird to have a virtual meeting and not meet face to face. And I do hope that you also enjoy the rest of the Congress. There are 2 other educational sessions prior to the real start. And I hope that there will be a lot of occasions to exchange and ask your questions. So thank you again, and goodbye.